All right, so utilizing all of the techniques that we have now gone over in chapter 12.1 and 12.2, we can kind of put it together as to what it means kind of on the whole, kind of back up from the molecular level, and then what does it mean as a whole. So we can use these things to study and compare genomes. And so we can go out and sequence. We can sequence, the fly's genome. We can sequence the human genome, and we can compare how closely related are a human and a fly, or different microbial species. So when we study and compare entire genomes, that's called genomics. So we can take a look at their complete set of genomes, or complete set of genes, their nucleotide sequence specifically, and how it's organized within that organism. And we can compare that to another organism, and that's genomics. One really popular genomics situation was the Human Genome Project. There were different companies that were all kind of fighting to sequence the human genome first. And so then we could use that information within the information that we had, or given the information that we had with the nucleotides, then we could do things like develop antibiotics, right? So if we know our actual gene sequences, then we know what's not us. And we also can know how something else might affect us. So we can develop antibiotics that are going to be then specific to those organisms rather than damaging our own DNA, for example. Or diagnostic tools, so things that can allow us to determine illness. And now that we know our DNA, we can do that better. Or vaccines, or different medical treatments, or environmental cleanup techniques, so we can determine how we can get rid of these different things, how we can clean up the environment based on the information that we have from the genomes. Now, if we move that over to our mRNA that we talked about as well, so in the entire collection of mRNA molecules that's produced by a cell is called transcriptomics. So if we are studying the mRNA molecules that are produced by cells, that's transcriptomics compared to genomics. So this we've been speaking about as well as we've been talking about these topics. So comparing between infected cells and uninfected cells. So what kind of cellular responses are we getting? Because if we're looking at the mRNA, remember that's just what's expressed. So not for genomics, which is looking at the genes, but we're looking at what's actually being expressed in an organism compared to another organism, or between two, infected or uninfected cells within an organism. We can also monitor gene expression of virulence factors in microorganisms, so we can apply different situations, different environments, those types of things, and we can see what is expressed, and by seeing what's expressed in mRNA molecules, we can see if those are virulence factors, and if we can increase or decrease the virulence factors given the environmental situation. If we then take either genomics or transcriptomics and we apply it to an entire microbial community, then that is metagenomics or metatranscriptomics, respectively. So if we want to, say, look at the microbial community in somebody's intestines, we can take that and then we can do maybe a DNA microarray on it, or we can go through the process of doing some labeling. And what we would be doing is we would be looking at the entire microbial community, because maybe we can't pick things out. Many can't be easily cultured, if at all, in a lab. So then what we can do is we can compare these microbial communities. So maybe we'll compare a microbial, intestinal microbial community from one person to another person, maybe on one side of the world to the other side of the world, and compare what their microbes are inside their intestines, and maybe relate that to food or health or something like that. Another type of study, given these kind of methods, these whole genome methods, is pharmacogenomics or toxicogenomics. And this is evaluating the effectiveness and safety of drugs. And this is on the basis of our individual's genome sequence. So first, we've done this on lab mice, so using animals in the lab. So lab mice or live cells in the laboratory. So meaning we can take a cancerous cell or a not cancerous cell, and we can apply something and see how toxic it is. The effectiveness of a drug. So if it's going to damage our cells and not kill off the bacteria or something maybe, then we can study that ahead of time. So using lab animals or using live cells. what we can do then is we can see the different changes in gene expression. So again, we would take, say, healthy cells, rather than saying a cancerous cell versus a healthy cell, and taking a look at the genome or the mRNA. Then what we can do is take two groups of healthy cells, we can apply something to one of them, say a pharmaceutical we want to test its effectiveness for, and then we can look at the mRNA, and then we can create a cDNA or something like that, and we can take a look at the effects. So in the end, here, someday, this information, what they're trying to do is make it be to where we can get an individual's genotype, and then we can make a specific drug based on that person's individual genotype so that it is the most effective and least toxic. So being able to sequence a person's genome and then create pharmaceuticals that will be helpful to that person. Most helpful, least harmful is kind of the end goal with pharmacogenomics. Then we have proteomics. So proteomics is studying the entire complement of proteins in an organism, or what's also called the proteome. So again, this is just saying like genomics is studying the DNA, transcriptomics is studying the mRNA and then the DNA made from the mRNA, and then proteomics is studying the proteins that they make. So again, which proteins are expressed under various conditions in a cell, a particular type of cell, or between different organisms. And then we can use this for seeing things like biomarkers. So what specific proteins are expressed during the disease process. So if somebody's diseased, we can take a look at what proteins the body is making. So then we can maybe get an idea of how it fights things off, or how it protects itself, things like that when a person is ill. Or things like this, used to detect cancer, for example. And so we can see the differences in the different proteins that we find. So we can also use some of these processes like the recombinant DNA or molecular cloning types of situations to do things like making antibiotics. So do a modification of the natural antibiotic pathway. We will talk a lot more about antibiotics in future chapters. But there are natural antibiotics and there are synthetic antibiotics, and then there are semi-synthetic antibiotics. And so some of the semi-synthetic antibiotics are going to be where we take a natural antibiotic and then we can modify that to make it work better or work on a different group. And so that would be done by a recombinant DNA process. So adding more DNA to it, for example, so that we can make a different type of antibiotic. And same thing with synthetic. We can improve yields or create new antibiotics through the introduction of genes. Also, subunit vaccines. So we'll talk about vaccines later, but there are subunit vaccines where we can make the different units so they contain only a single antigenic molecule, and then they would lack part of the genome of the pathogen, but then we can insert the gene that codes for a particular protein. So an example, we can insert the gene coding for hepatitis B, surface protein, into yeast. And if we do that, then we can inject yeast into a person that has this particular gene on it, rather than injecting hepatitis B, for example, and we would make a vaccine that way. So we would purify it and then inject it as vaccine, and then the body can produce antibodies against hepatitis B, but it's not because they've been given hepatitis B, it's because that actual information, that gene, is going to be placed onto yeast, and then that would be injected into the person instead of actually taking a chance at hepatitis B. And then also insulin. We've already spoken about this quite a bit. We're using E. coli and then putting the gene for insulin into it, and then we make lots of copies of E. coli. It doubles every 20 minutes, so we get lots and lots of insulin that way. So these are just some of the ways that we can take these processes that we've just spoken about and then apply them to kind of things that are happening every day. So making antibiotics, making vaccines, making insulin for diabetics. All right, so the next thing we are going to look at is RNA interference technology. So before we look at the actual technology part, or the biotechnology part, let's take a look at what RNA interference is, because we haven't spoken about this yet. So RNAi, or RNA interference, is something that happens within a cell already. It naturally occurs. And this is where we have different mRNAs that are going to attach to an mRNA that's being transcribed, and it's going to prevent it from being translated into proteins. So these mRNAs are prevented from guiding the synthesis of proteins. What's going to attach to it is the antisense RNA. So the antisense RNA is going to be complementary base pairs to this particular mRNA. And so these are naturally occurring because they are there to protect against viral RNA. So remember, if we have a single-stranded RNA virus that comes into the cell and tries to hijack things, it's going to have to get its RNA out there. And if we have any of these antisense RNAs that are hanging around that complementary base pair, then it can stop the ability of that virus from taking over the cell and then taking over the machinery and causing infection. So we have these antisense RNAs out there. They complementary base pair to mRNAs, and then what happens is we have this double-stranded RNA molecule, and the double-stranded RNA molecule cannot go through translation, and so translation doesn't occur. So if it's coming in from a virus, we have our single-stranded RNA, and then we have our antisense RNA that anneals to it or attaches to it. Then it stops the virus from being able to make copies of itself or any copies of its RNA. So as far as our technology is concerned, they are trying to use this to develop ways to treat viral infections. So if we can somehow get a known specific antisense RNA into the cells or the body of a person that is infected with a viral infection, then we could perhaps cause this double-stranded RNA to be formed, and then the cell, or rather the virus, isn't going to be able to make copies of itself. You know, the virus using the cell wouldn't be able to make copies of itself. And so then we could treat viral infections that way. We also use this RNA interference technology currently in something called gene silencing. When we are going through the process of gene silencing, we're utilizing either small interfering RNAs or siRNAs or microRNAs or miRNAs. The difference between these two is that our small interfering RNAs or siRNAs are completely complementary to a gene sequence, versus our microRNAs are mostly complementary to a gene sequence. So maybe we don't know the exact gene sequence of this particular viral DNA or this bacterial DNA. So we have a microRNA that's mostly complementary versus if we know the genome, we know the makeup of a particular pathogen that we're trying to get rid of maybe, we can get our complete complementary gene sequence. So we can do an siRNA. So how this works is kind of illustrated here on the right side. We have our RNA molecule, and that's going to bind to dicer. Dicer is an enzyme. So it's going to bind to dicer, and then it's going to be cleaved into short molecules. So it's going to be cut up into small molecules. So then depending on the structure, depending on the nucleotide sequence, we either have our miRNAs, which are mostly complementary, or we have our siRNAs, which are completely complementary. So then we have our miRNAs or our siRNAs are going to then bind to RISC, R-I-S-C, which is another enzyme, which is our RNA-inducing silencing complex. So it's multiple proteins put together. So this complex complementary binds to the mRNA. So it's going to kind of hold on to this mRNA. Then what it's going to do is it's going to find this complementary sequence. So our RISC enzyme is going to have this attached to it. Then it's going to be floating around, and when it comes upon our RNA, that it can either partially bind to, because remember this is imperfect, or that it can completely bind to, like in the siRNA. Then what it's going to do is if it's the siRNA and it's completely bound to it, it's going to cleave it. So it'll cut that mRNA. If it is miRNA and it is not perfectly bound because it's mRNA, then it's just going to stay bound to that RNA molecule, that mRNA molecule, so it just can't be transcribed. So in the case of our siRNA, it's going to cut up the mRNA so it doesn't make the proteins that it's meant to make. With the miRNA, it's just going to stay bound to it so it can't make the proteins anyway. So with this gene silencing, this is one of the ways that scientists are trying to, again, get rid of infections, get rid of viral infections or bacterial infections by using these small interfering RNAs or the microRNAs, utilizing gene silencing. So it's stopping the ability for the gene to be able to end up making a protein. So then lastly, in 12.4, there's a little section that talks about gene therapy. So this is our clinical application of genetic engineering. So we did speak a little bit about this already, where we're trying to change the DNA in a person's cells, for example. So we mentioned before we could do this perhaps with viruses, where we take out the viral DNA, particularly the virulence proteins, and we can then put in eukaryotic DNA or our DNA, and then hopefully that will then incorporate itself into the chromosome. And then once it's incorporated into our chromosome, one of our chromosomes, then we have effectively changed our DNA or adjusted the DNA. And that is kind of the basis for gene therapy. So we can use an adenovirus vector, and then we're using it to either try to treat or cure genetic diseases, right? So if somebody passes down a genetic disease, what they're trying to do is use gene therapy. So the patient has this mutated or defective gene that causes their genetic disease. Then we can try to correct that by introducing a non-mutated or functional gene. So utilizing this adenovirus. So you can see here the vector binds to the cell membrane, and inside of it what we have is something we've created. So we have the viral DNA and both sides, and then we have our new gene. So this would be the non-mutated or defective gene for a particular, whatever that gene codes for. So we've created this. We have a brand new gene that's not that's not mutated, and it's in with the viral DNA. So again, we take out the virus part. Basically, what I mean by that is the virulence piece that causes infection. And then what we do instead is we allow it to have all the rest of it, where it's going to go in and kind of hijack things and incorporate its own DNA. But of course, in this case, it would be incorporating the correct version of a gene. So it binds to the cell membrane, packaged in a vesicle, comes down in, it breaks down, releases this, and then the vector is going to inject the new gene into the nucleus, so that now the cell is going to make proteins using this new gene, because it's just going to go along and it's going to make proteins like it normally does, transcribing and then translating, but now it's going to be making an effective enzyme from this, or an effective protein. So this is really neat, very cutting edge, but there are some risks and some ethical concerns, right? So some of the risks is that it could trigger the inflammatory response. So in somebody that already has a disease, for example, maybe they're immunocompetent, and so then doing this gene therapy could really put them in a bad spot, because even though it might not have the virulence genes, if we're still putting a virus in the body, it might have a response and then could cause organ failure, for example, because the body just can't handle it because it's already immunocompromised. Some risks: the infected cells could not be targeted. So if we do put this in, remember, we can't really determine what cells are going to be infected. So if we try to go in there, it might not end up infecting the correct cells anyway. So then it could be damaging, it could be infecting other cells and then messing up how those cells function, or cancer causing, right? Because if it's going to be messing with the DNA and it doesn't go into the right cells, then it could start to cause cancer in various areas. Some other issues, and we see this with vaccines as well, is that the virus could revert to being infectious. So it can modify and change its DNA. It could modify and change its DNA while it's inside of the person. So then this could end up causing disease. It could go back to being an infectious bacterium or virus, rather. It could inactivate another important gene. So, for example, it could incorporate itself inside of a really important gene, and then that could cause worse. So it could cause tumor formation, it could cause cancer. It could end up being really bad if it incorporates itself into a very important gene for a very important protein. So then we also have the ethical concerns. So you can see, is it possible, or should we be able to go in and adjust these types of things? And we can talk more about the ethical concerns and watch some videos based on some genetic engineering and take a look at the pros and the cons in class as to what might be, we just spoke about the risks, but then ethically, is this the right thing to do? Can we do this and perhaps it's going to cause more disease? So like in the person where it might inactivate another important gene, right? Can we ethically sit down and say, look, like we know that you have this genetic issue here, this genetic disease, and we want to inject you with a virus that will hopefully, fingers crossed, get to the right cells to then incorporate itself not in an important gene, but in a non-important area, non-coding area that then your body will hopefully start to make proteins from and then fix that genetic condition. So there's a lot of kind of hoping going along in that statement. And so there are a lot of ethical concerns related with that. Right now, there's significant oversight of these different gene therapies at the federal level, some local levels. So, for example, some of the people that are overseeing this are the Food and Drug Administration, the Office of Human Research Protection, Recombinant DNA Advisory Committee, and National Institutes of Health are all kind of working on gene therapies right now. And some of them seem to be going very, very well. But it's slow moving, especially because of these ethical concerns.